Imagine a mirror world. It looks just like ours, but everything is flipped. Your left hand becomes your right, and a clockwise spinning top spins counterclockwise. This is the world of chirality, where molecules, like our hands, exist in two forms, mirror images of each other. In the vast universe of molecules, many exist in these left-handed and right-handed versions. We call these molecules chiral. It's like looking at your hands. They are mirror images that can't be superimposed on each other. Now imagine life's building blocks. The molecules that make up you and me also have this curious property. Amino acids, the ingredients of proteins and sugars, the fuel of life, can exist in both left and right-handed forms, but here's the twist. Life on Earth has a distinct preference for one hand over the other. We call this fascinating phenomenon biological homochirality. Life on Earth has made a choice. It has chosen left-handed amino acids to build proteins, and it uses predominantly right-handed sugars. This is like walking into a bakery and finding only croissants curled to the right, never to the left. It's a puzzling asymmetry in the otherwise balanced laws of nature. Why is this important? Because this choice of chirality has profound implications for the very existence and functioning of life as we know it. Imagine trying to build a house with mismatched bricks, some left-handed and some right-handed. It just wouldn't work. Similarly, life relies on the precise fit and interaction of molecules, and that fit is determined by their chirality. How did this preference for one hand arise? Scientists are still trying to solve this fundamental mystery. It's like trying to decipher a message from the early universe, a message written in the very molecules of life. Some believe that the answer lies in the conditions of the early Earth. Perhaps a slight excess of one type of amino acid due to some external factor like polarized light from a distant star tipped the scales. Others propose a more internal mechanism, a kind of molecular domino effect. Imagine a single chiral molecule replicating itself, creating more of its kind, and setting off a chain reaction that ultimately led to the dominance of one hand over the other. Regardless of the origin, this early choice has echoed through billions of years of evolution. It has shaped the very fabric of life, influencing how molecules interact, how proteins fold, and how life itself functions. Imagine a perfectly fitting glove. Your left hand fits snugly into the left glove, but not into the right. This is the essence of molecular recognition, a fundamental principle in biology driven by chirality. Proteins, the workhorses of our cells, are built from these carefully selected left-handed amino acids. They fold into intricate three-dimensional structures, their shapes dictated by the precise sequence of these amino acids. These shapes are crucial. They allow proteins to interact with other molecules with exquisite specificity, like a lock and key. A right-handed protein simply wouldn't fit into the intricate machinery of the cell, disrupting the delicate balance of life. This chirality-driven specificity is everywhere in biology. Enzymes, the catalysts of life, use it to bind to specific substrates, facilitating chemical reactions. Hormones recognize their target cells with the same precision, ensuring that messages are delivered to the right place at the right time. Section 5. Shape-shifting proteins. Proteins are the workhorses of our cells, carrying out an astonishing array of tasks. They transport molecules, catalyze reactions, provide structural support, and much more. But proteins are not rigid statues. They are dynamic, constantly moving and changing their shape in response to their environment. Some proteins, known as allosteric proteins, take this shape-shifting to a whole new level. Imagine a protein as a complex origami structure. Now imagine being able to fold and unfold certain sections of this origami by simply pressing on a specific point. That's essentially what allosteric proteins do. These proteins have specific sites called allosteric sites that act like molecular switches. When a molecule, called a ligand, binds to an allosteric site, it triggers a change in the protein's shape and therefore its function. Section 6. The choreography of change. How do these proteins know when to change their dance moves? How does the binding of a molecule at one site affect the shape and function of a distant site? It's all about the intricate choreography of molecular movements. There are two main models that scientists use to explain this allosteric regulation. The concerted model, also known as MWC, and the sequential model, or KNF. The concerted model, like a synchronized dance troupe, proposes that proteins exist in two preset conformations, active and inactive. Ligand binding simply shifts the equilibrium between these states, like a conductor cueing the orchestra to switch melodies. The sequential model, on the other hand, is more like a wave traveling through a stadium. Ligand binding induces a local change in protein shape, which then propagates through the molecule, ultimately affecting distant sites. The reality is likely a combination of both models, a complex interplay of pre-existing equilibria and induced fit. But regardless of the mechanism, the outcome is the same precise control over protein function. Section 7. Life switchboard. These shape-shifting proteins are essential for life. They act like molecular switches, regulating a vast array of cellular processes with remarkable precision. One of the most well-known examples is the regulation of enzyme activity. Metabolic pathways, the chains of chemical reactions that sustain life, are carefully controlled by allosteric enzymes. These enzymes can be switched on or off, sped up or slowed down, by the binding of specific molecules. This ensures that the cell produces the right amount of each molecule at the right time, avoiding wasteful overproduction or harmful deficiencies. Allosteric proteins also play a crucial role in signal transduction, the process by which cells communicate with each other and respond to their environment. Imagine a chain of falling dominoes, each domino representing an allosteric protein. A signal, like a hormone binding to a receptor on the cell surface, triggers the first domino to fall, initiating a cascade of conformational changes that ultimately leads to a specific cellular response. Section 8. Unlocking nature's secrets. By understanding these molecular dances, we can unlock nature's secrets and develop new ways to treat diseases. Many drugs target allosteric sites, modulating protein activity with greater specificity than traditional drugs that bind to the active site. This opens up new avenues for drug discovery, allowing us to target previously undruggable proteins. For example, imagine designing a drug that doesn't directly block an enzyme's activity, but instead fine-tunes its activity by modulating its allosteric regulation. This could lead to more effective treatments with fewer side effects. Understanding allosteric regulation is also crucial for unraveling the complexities of diseases. Many diseases, from cancer to Alzheimer's, involve dysregulation of allosteric proteins. Section 9. The cosmic connection. The universe is full of mysteries, from the vastness of space to the intricacies of the quantum world. But sometimes the most profound mysteries are found right here on Earth, within the very cells of our bodies. Biological homochirality and allosteric regulation are two such mysteries. They remind us that life is a delicate dance of molecules, a dance choreographed by billions of years of evolution. By unraveling the secrets of this molecular dance, we gain a deeper appreciation for the complexity and elegance of life, and we open up new possibilities for understanding ourselves and our place in the cosmos.